Thank you. Good afternoon. I'll tell you a little bit about a project that I've been working on as a postdoc at the University of Strathclyde. So, teeny bit of history. We started with a tool called pyANI, which is short for Python Average Nucleotide Identity, that the group had already written, and then got grant money to re-implement, to redo it and do it a bit better this time around. So average nucleotide identity is used to compare microbial genomes, particularly bacteria. It's an embarrassingly parallel problem because you compare each pair of genomes independently of the others. So naturally, you're going to want to run this on a cluster once you get up to size. Just to note, we had lots of contributions from the community on the old version, and that included me as an external member of the team. And this time around, we're trying to do it better. We were already practicing most of the good engineering things that you would be doing. We had open source licensing, public repository, continuous integration, documentation, pre-commit hooks. Hopefully everyone in the room knows what those are. When you save a snapshot of your code, it checks the obvious things like, does it compile? Are you following the style guidelines? It keeps your code clean as you go along. This time around, new things, we're using something called type annotation, which is an optional feature in Python that records what a data type should be, and it indirectly catches a whole bunch of errors. And we have full test coverage, which I'm quite pleased and proud about. A couple of corner cases for race conditions and interrupts that really couldn't work out how to test, but otherwise full coverage. So we took the old code, we've reworked it. We've added a few new methods. One is called SOWMASH. It's a very fast KMA-based approach for comparing genomes. And a way of importing an external alignment. We've got a poster D094, which will go through the scientific applications of this. If you've got a synthetic ground truth, you can actually evaluate how the different existing methods work on your synthetic data set. And what the community may have thought was the best method isn't necessarily the best method. But I will save that for the poster. The original integration with the cluster was done with the SGE cluster system with a standard Python library. We had to write that and maintain that, and it was a burden. And then our cluster moved over to Slurm instead, so we couldn't use the cluster anymore. This time around, we are leveraging SnakeMake. So SnakeMake is normally used as a workflow system, but you can call it as an API, and we're using that internally. And SnakeMake can talk to various cluster systems. And in particular, it talks to Slurm, and that's what we've used it for. It's tested on 2 different Slurm clusters. On the technical side, we're doing potentially millions of comparisons, and we store them in a database. We're using SQLite 3 because it's really simple to install as a dependency for the end user. You don't need a DB admin. There is a complication when you want to write your data to the database. When you've sent this out to a cluster and you've got lots of different compute nodes, they've all got results, they want to put them in the database. We tried initially the naive approach, database on a shared network drive. You run into locking problems quite quickly, unfortunately. We tried with that a bit, but we gave up. The solution that works really well for us is that each of the workers does the computation, passes the output files, and records just the bit of information that needs to go in the database in a JSON file. Back on the master thread on the worker computer, that monitors for the new JSON files, imports those into the database. So you've got one thread doing the writing, and it can update a progress bar for the user as it goes along. And that has scaled beautifully. We've run it with thousands of workers at the same time. You need about a day to use the slower methods on 1,000 bacteria. An example plot, we're using standard Python libraries, matplotlib and seaborn. One of the weaknesses of the old tool was dealing with missing data. So when you compare quite diverged genomes, some of these genome comparison tools just give up. And they say no, they fail. So we get non-applicables or nulls in the data. Those are shown now as orange in the heat maps. And that can be quite instructive just where all the orange bands show up. The red-blue colors is a traditional color scheme for the genus-species boundaries that people accept based on the literature. You can be a bit more systematic now. This is a quite complicated new plot that we came up with. You can vary your threshold. You don't have to pick 95% as your species threshold. And you can see if you cut off at that point, what natural groups form the data. And you get cliques that may indicate biological functions. Sometimes it matches beautifully to genus and species, Other times, what comes out of the data doesn't match the taxonomy. There's a big pretty picture, that's 1,000 pectobacteria. We're using Python libraries typer and rich to make a pretty command line interface. I've been quite impressed with those tools. Acknowledgements, we've used the Archie West cluster in Strathclyde and the crop diversity cluster at the Hutton Institute. Funding was through BBSRC. Thank you very much.